Toxciology for the introduction. Today I will be very glad to present our work on the applications of CFD to investigate the inhalation toxicology of inhaled nanoparticles into human airways. And today's talk will be focused on the nasal olfactory. So humans breathe all the time, and at each breath, particles of different sizes, different shapes can come into the airway. And those particles will interact with the airflow as well as the geometry. It might deposit at different locations and have a different fate. Some will be expelled through the airways through mucus. Some others might penetrate deep into the airway and translocated into other parts of the body. One of the notorious examples is asbestos. So asbestos is elongated, needle-like shaped particles, which can penetrate deep into the airway. There is no effective self-defense system which removes these particles from the airway. As a result, they can accumulate and come across the respiratory barrier and lodge into the cavity. The problems with this type of accumulation is that you don't see the symptom right away. After 20, 30 years down the road, when symptoms occur, it's always too late. In recent years, there are growing interest, growing concerns about another uptake, another pathway to uptake particles, nanoparticles through olfactory pathway and translocate it into the human brain. The problem with this type is also because it's usually very low dose. We don't see the symptom right away. When we notice there's already damage. The mechanism is to explain if we look about autonomy. So for every breath, we're conducting the air from the nasal cavity. A secondary function is olfactory. So a small amount of molecules might will come into contact with the olfactory receptor neurons at the layers in the nasal side, and they will actuate the neurons to transport the signals through the nerves into the olfactory bulb. Olfactory bulb is at the brain side, which further have neural networks connected to the central nervous system. So it is clear that smell can come from the air into contact with the neurons, passing through this route and into the nervous system. So is there possibility that for particles, nanoparticles, which if contact or deposit into the nasal cavity side, can it also take advantage of this route translocating into the brain? Findings from the relationship between Walding and Parkinson's disease shows that it's very likely there might be such a route. Walding fumes are a mixture of metal oxide at nano and micro scale. They have different shapes. Frequently, they contain magnesium, which is highly neurotoxic. So if we have acute exposure, we have acute poisoning. But what happened to subclinical exposure, in which case we don't see symptoms right away? But preliminary findings find that for riders, they have a higher risk of developing Parkinson's disease at an early stage. So for Parkinson's disease, what happens is they have, for the patients, they have degenerated neuron cells around this region, which is right behind the olfactory. And is it possible to come, for the particles to come into the nasal olfactory through another routes? The blood-brain barrier, the findings of the blood-brain barrier, which has a very tight conjunctions, prevent the entering of nanoparticles, for example, magnesium, into the brain through circulatory system. But for wilders, what they find is they have an elevated presence of magnesium in this region. So where it comes from? It's highly likely from the olfactory. So there's a very interesting experiment. It's done in 1997 by this group. What they do is they inject magnesis through the nasal cavity for rodent. What they did is they cover one side of the nose. They inject through only one nostril. Over the time, they detect penetration of this magnesis through the olfactory barrier into the olfactory bulb in the brain side, and eventually enter the brain. What's interesting is the other side of the brain is unaffected. So it is clearly shown that this penetration and translocation of the particles is through nasal olfactory pathway. What happened to human? So there's another preliminary experiment done in 2011 on human volunteers. So what they did is they inject thathium into human nasal cavity. 30 minutes after injection, they detect the concentration of this thathium is only in the nasal cavity side, which is the red circle. 24 hours later, they detect the concentrations of thathium into the brain side, which is an olfactory bulb. What happened next, we don't know, but this clearly shows that it's very likely what happened in human is probably similar to what happened in the rodent. So olfactory pathway for brain uptake of inhaled nanoparticles is possible, is highly likely, and is a sequence of events. First, inhalation into the nasal cavity, and then deposition into the nasal olfactory, and then penetration into the nasal olfactory bulb in the brain side, and the further penetration or translocation into the brain. So we need to understand each stage carefully before we completely understand the entire process. For our study, we would like to focus on the first stage, which is inhalation into the nasal cavity, deposition into the nasal olfactory. So here's the study. A study focused on computation modeling of transport of inhaled nanoparticles to human olfactory, and then identify the mechanism. So what affect the deposition? The deposition pattern, deposition efficiency, the quantifications, and then those deposition relationship. And then finally, we want to know human-rat correlations. So here we apply the methodology which has been presented earlier by many people working in this field. So we obtain medical scans, reconstruct the 3D nasal airway model. We label the nasal cavity with different regions, and in this study we focus on number 3, which is the olfactory region. This is the cross-sectional area. We can see the olfactory is in the ceiling, occupy a very small portion of nasal channel. He then developed this code which can unwrap the 3D surface model, which is human. This is rat. We can unwrap this 3D model into 2D surface map. Why to unwrap it? If we want to study the surface features, for example, the shear stress, deposition patterns, the 2D gives better visualization. With all in place, we're ready to apply the fluid simulation. So here I want to repeat again, we have the 3D model, nasal cavity. We identified the olfactory region. We got a cross-sectional visualization, and we unwrap it in 2D space and identify the olfactory, which is located in the occupied a very small region of the entire nasal cavity. Here I mention again because there's no experiment study in human. Most of the study, even if you want to investigate, will be performed in rat or animal study first. So it's necessary to understand what happened to rat as well as what happened in human. and do meaningful interpolations of the data. We apply the same method, exact same method again, to get the 3D nasal cavity model for the rodent, for the rat, and then identify the nasal olfactory for the mouse, rat. And this is cross-sectional channel, and this is the 2D unwrapped of the nasal cavity. And this area shows the olfactory for the rat. So we apply computational meshing the geometry. We use polyhedron meshing. Fluid simulation, then apply fluid simulation. It's as you can see, this continuation momentum equation, we are considering sedentary, which is low velocity, is laminar flow. So airflow simulation results, it can give us a direct visualization about how the flow is distributed in the nasal cavity. In this study, what we care or focus is about the olfactory, which is on the roof. As we can see, the flow is very low. We will expect a very minimum volume of airflow reach this region. And this is nasal wall shear stress. This 3D presentation is 2D presentation. Nasal wall shear stress has an indication about the secondary flow in the nasal cavity, which is closely related to the eddies. The particles might follow these eddies, and it's related to, also has indication about the deposition. So this information will be also helpful for us to analyze the physics in the cavity. Then we inject the particle. The particle we consider as a discrete phase. Discrete phase of the particle is modeled as Newton's second law. We consider the drag, hydrodynamic drag, gravity, lift force, and Brownian diffusion. So we can integrate those equations and reproduce what happened to the inhaled particles. We'll be able to reproduce what their trajectories in the nasal cavity. We know how many of them are deposited and where they are deposited. We can compare what happened in the rat, what happened in human, and we also be able to analyze the effect of size of the particles. This shows a 1 micrometer particle deposition simulation results. We can also, for different flow rate, we can also have different comparing metrics. This is for 5 micron. Most of the particles, they cannot get across the nasal frontier in the rat, but for human, we have still quite a large amount get into the nasal cavity. And this is a 1 nanometer trajectory. This is 100 nanometer trajectory. So this information can give us a qualitative comparisons analysis of what's happening of the particles motion inside nose, where it's going to deposit it, and where it's going to deposit and what parameters are affecting them. This is another parameter, particle flux. Particle flux is defined as number of particles passing through per unit area of nasal cross-section area. It's normalized. Basically, the indication shows how many particles is distributed across the channel, nasal channel. This is the simulation result for particle flux across nasal channels. This is the nostril, nasal valve, and mid airways, and this is pharynx. This is for 1 nanometer size particle, 2 nanometer, 10 nanometer. So it's clearly seen that the particle distribution is affected by the particle size. For 1 nano, we don't have much flux in the nasal olfactory. It's increasing for 2 nanometer, and for 10 nanometer appears to have more flux. And what we focus, the simulation, CFD simulation can give us information. We can find the preferred particle distribution in correlation with the airflow pattern. We can clearly see the particle size effect which is related to molecular diffusivity of the particles. And we can also focus on olfactory particle distribution. And this is a particle deposition pattern. This is a 2D surface map. This inner area is the olfactory. The red dot means the particle deposited in other parts of the cavity, and the blue one means the particle deposited in nasal olfactory. So it's clearly seen that the deposition pattern in the nasal olfactory is very different for different size particle. For 1 nanometer, it's all concentrated into the border, near the border of the nasal olfactory. And then at 2 nanometer, it penetrates in. And at 10 nanometer, it's more homogeneous. So which clearly shows different deposition mechanisms. And also very important information, is the sensitivity of deposition of small nano-sized particles into nasal olfactory. Because nasal olfactory surface area is not well defined, I look about the literature, didn't find definite answers about what's the border, what's the size. And there might be a variety between different people. And in literature, I even see studies trying to double the size of the nasal olfactory, half the size of nasal olfactory, trying to find the deposition, trying to find the difference. So this clearly shows that if the size varies in human about the nasal olfactory surface area, the deposition could be completely different. Could be significantly different if the size shrinks or enlarge a little bit. So this can give us a lot of information we can further investigate. And finally, the particle deposition efficiency. It gives a quantitative measures about how much particles deposit into olfactory region. So its deposited particles really divide the particle internal nasal cavity. So here's the particle deposition efficiency in the entire nasal cavity for different flow rate against different diameters. This is the same information, but we plotted at a different at a combination of the parameters. This is D as diffusivity and Q as flow rate. So apparently deposition proportional to diffusivity, but inverse proportional to flow rate. We plot our results with experiment data, which is scattered over here, and the simulation from prior researchers. The trend agrees. The trend agree. We also formulate our empirical equation. We can predict deposition efficiency in the nasal cavity quickly if we know the size, we know the flow rate. So for olfactory deposition in human, we can see the trend is completely different from nasal cavity in the total deposition in nasal cavity. So we cannot use the same equation or the same form equation to predict the deposition. We see that the olfactory deposition is low for extremely small particles, less than 1 nano. The peak occurs around 2 or below 2 nanometer, and then quickly drop. The flow rate also affects the olfactory deposition significantly for smaller size particles, but for larger ones, the significance is not that obvious. Apparently, compared with total deposition, we see here is 3.5. So the deposition rate for olfactory is very low, and also it's sensitive olfactory size. Another very interesting observation is we plotted the particle deposited into the nasal olfactory as blue. So these are the nostrils. So the particles all enters the nostril cross-section in this pattern. But for the particles deposited into the olfactory, they are located at this area. So for particles, if at the bottom or lower half of the nostril, there is no way that it can deposit in the olfactory. Only particles in this top corner, it can enter the olfactory. And for 10 nanometer, it's very small region of the particles. If they just enter in this region, they can reach the olfactory. That might be the reason when you smell something, you have to smell it near your nose tip and smell it increase, just sniff hard in order to get those particles into the smelling zone. So this is future work. We have just studied what happened inhalation into nasal cavity, deposition into nasal olfactory. But what happened afterward? How it is penetrated through this region, how it's further translocated into the brain is still open for investigation. There are very interesting congestions. One hypothesis is the nerve itself can grab the particle, and then somehow this nerve can transport those particles into the brain. But I don't know if that's correct. For me, it makes more sense if those particles can penetrate through the junctions, and then transport through some openings right here. But these are completely new, still unknown, and still need future investigation. So this is my talk, and thanks for your attention. Any questions?